Okay, this video looks to explain how the neuromuscular junction works. So what we are looking to understand is what happens when action potential arrives at the end of an axon which is connected to a muscle fibre. The image on the screen shows the neuromuscular junction. On the left hand side you have the end of an axon, and on the right hand side you have a muscle fibre, and you can see how the sarcolemma has been folded to form T tubules which go deep inside the muscle fibre. So when an action potential arrives, as it's about to, at the end of the axon, this triggers voltage gated calcium channels in the end of the axon to open, and calcium ions will diffuse into the end of the axon. The presence of calcium ions triggers vesicles which contain acetylcholine to move towards the end of the neuron and basically fuse with it and release the contents via exocytosis. So the vesicles fuse with the membrane at the end of the neuron and release the acetylcholine into the gap, and the acetylcholine will diffuse across the membrane and bind to receptors which are found on the membrane of the muscle fibre, the sarcolemma. When they bind to receptors, the sodium ion channels which the receptors are found on are triggered to open, and sodium ions which are found in the gap will diffuse through the sodium ion channels into the muscle fibre. This depolarizes the membrane, and this depolarization sweeps like a wave down the T tubules to the cytoplasmic reticulum. When it gets to the cytoplasmic reticulum is triggered to release calcium ions, and this triggers muscle contraction. This is another diagram summarizing what we've just been talking about. Neuromuscular junctions and synapses have quite a few things in common. There's a few differences though, and it's a common exam question to be asked to compare the two. Neuromuscular junction, the neurotransmitter will always be acetylcholine, whereas in a synapse between two neurons it can be a variety of different neurotransmitters. It can be acetylcholine, but it could also be perhaps dopamine or serotonin. The postsynaptic cell in a neuromuscular junction will be the muscle fibre, whereas after a synapse you will find another neuron. The presynaptic neuron, the shape of the end of the axon, for a neuromuscular junction it's described as having a brush border, whereas the presynaptic neuron is smooth and round. It's sometimes called the presynaptic knob. The effect of the action potential on the presynaptic neuron is the same in both cases, where calcium channels are open and the calcium ions flood in, when this triggers vesicles to be released into the cleft. The effect of the neurotransmitter binding on the postsynaptic receptors is also similar. They open sodium channels and they will flood in, or the sodium ions will flood in, and this depolarizes the membrane. In the case of a neuromuscular junction, the depolarization will sweep down to the T tubules towards the sarcoplasmic reticulum, and when it gets to the sarcoplasmic reticulum it triggers calcium ions to be released from it, and this triggers muscle contraction. In both cases it is an enzyme which will break down the neurotransmitter. The specific enzyme to break down acetylcholine is acetylcholine esterase.